The topic of my talk today is introducing a new concept known as neurogenesis hypothesis. We use this one, a case study of our drug Phase II trial of our drug NA-831 for the treatment of Alzheimer's disease. Virtually all failed Alzheimer drugs of the past 30 years were designed mainly to remove the amyloid plaque, according to the hypothesis, as we were aware. To my opinion, the amyloid hypothesis is an unproven hypothesis, which proposed that the Alzheimer's disease is caused by the buildup of the amyloid beta plaque. Recently, as you know, Biogen drug aducanumab has been approved by the FDA in June 2021. But if you look closely, this drug does not show noteworthy improvement of cognitive functions in Alzheimer's patients. And the toxicity was so severe, with more than 42% of the subjects participate in 2 Phase III trials develop ADR, which is the amyloid-related imaging abnormalities. It is a brain swelling and aseptic meningitis. Some of them could be a fatal condition. And on top of that, lecanemab was developed by Eisai and Biogen has been approved by the FDA on January 2023. And once again, both drugs, aducanumab and lecanemab, were based on the surrogate endpoint biomarker, the reduction of the amyloid plaque in the brain. And I think it is a very promising for all of us in the Alzheimer research community and the patient community that at least there are 2 drugs have been approved by the FDA. And it is the beginning of a long journey, I believe, and it brings some hope. However, these drugs were not shown clear clinical benefit for the patients. As a result, Medicare or CMS declined the reimbursement of aducanumab, and recently they also declined the reimbursement of lecanemab for majority of Alzheimer patients, except those patients enrolled in additional clinical trials. This is some setback for Biogen and Eisai. And I think all the non-profit organizations have tried to appeal to CMS to open the gate. And I think the decision is still pending. And so far, we accept the fact that both drugs, aducanumab and lecanemab, have not been accepted by CMS of the US, and these drugs have not been approved for reimbursement. So, understanding the environment at Biomed Industries, we would like to present a new pathway, a new hypothesis known as neurogenesis hypothesis for Alzheimer's disease. Most of us know that the hippocampus, being critical for learning and memory, continues to generate new neurons throughout life, a process known as adult hippocampal neurogenesis. The reason we call adult neurogenesis because, as you know, when we are young and a human being in the early infant development, our brain continues to generate it, and it is the neurogenesis. But we thought, and scientific research over the past many years will let us believe that when we reach a certain age, being adult, we no longer have the ability to generate new neurons in the brain. And that thought will actually prove to be not correct. Research shown that hippocampal neurogenesis is persistent throughout even the 10 decades of human life, meaning up to 100 years of age. And It is detectable in patients with Alzheimer's disease and with the people without the disease. Nesting SOX2 plus neuroprogenitor cells and DCX plus neuroblast and immature neurons were detected in subjects at 90 years of age, according to research paper by a research team Moreno, Jimenez, year 2019, and Tobin and the team at in the year 2019 as well. And it was observed that there was a marked and progressive decline of DCX cell numbers in Alzheimer's patients as compared with neurologically healthy individuals. So when the adult hippocampal neurogenesis become impaired, it would compromise the hippocampal functions in Alzheimer's disease and also mild cognitive impairment. Adult hippocampal neurogenesis impairment appears to take place prior to the presence of senile plaques or even before the presence of neurofibrillary tangles and FS, and even before we're able to detect the presence of amyloid beta. And these are thought to be the key pathologically hallmarks of Alzheimer's disease, particularly in the dental gyrus. In other words, even before amyloid beta can be detected, the certain phenomena going on, the adult hippocampal neurogenesis went through the period of impairment. As a result, the patient begin to losing the memory and cognitive functions. So we believe that the hippocampus is the main focus of Alzheimer's disease because it is the center for learning and memory. If anything happened to the hippocampus, it causes significant impairment for the patients, become dementia and Alzheimer's disease. So we believe that stimulating the inherent adult hippocampal neurogenesis could serve as a therapeutic target for improving cognitive functions and improving synaptic resilience. And this is the basis of our hypothesis. And based on hypothesis, it has guided us to develop a new drug over the past many years. We call it NA-831. This drug is a small molecule drug. It easily crosses the blood-brain barrier with great bioavailability. It exhibits both neuroprotection and neurogenesis. Now, how does it work? We found out that the drug NA-831 serves as a catalyst to restore neurogenesis in Alzheimer's disease patients by increasing DCX+PCNA cells to increase the number of neuroblasts, and therefore it led to the improved cognitive functions. This is our hypothesis. And there is a statement by Professor Nicolas Tony of Lausanne University in Switzerland. He mentioned that even if drug companies could develop therapeutics that block or reduce the amyloid beta, they will not reverse the damage. But adult neurogenesis in the hippocampus could be a way to partially right that wrong. I think that is a pretty good statement. In other words, a lot of research focuses on amyloid. We believe that amyloid beta already in the later stage. And that's why many clinical studies have not shown a clear benefit, even though we go further and test the drug for people with early phase of Alzheimer's disease. It might be a little bit too late because the amyloid already there it is irreversible damage. Okay, so by looking to further down, we believe that the neurogenesis is a viable target for us to consider in our research. Let me summarize the Phase IIa study in subjects with mild cognitive impairment and subjects with mild and moderate Alzheimer's disease. It is a randomized clinical trial of the drug will perform on 32 patients with MCI and 24 patients with mild and moderate Alzheimer's disease. The cohort is small, but it is the first time of the drug in human patients. So we like to just to demonstrate a proof of concept under the Phase IIa randomized placebo control. And the patient with MCI received 10 milligrams of the drug or placebo orally per day. Patients with the mild and moderate AD received 30 milligrams of the drug or placebo orally. Because this is a randomized placebo control, we have to have one cohort with the drug and one cohort with the placebo. So the primary outcome measure were the changes in Alzheimer's disease assessment scale, cognitive subscale, ADAS-Score 13 element. We also use the brief cognitive rating scale, BCRS, and clinician interview-based impression of change plus caregiver input, CIBIC plus, after 24 weeks. The result can be summarized as follows. The drug NA-831 shows significant improvement for MCI patients with a ADAS-Score 13 scale change of an average of 3.4 as compared to placebo after 24 weeks of treatment. The p-value equal 0.1 ITT. NA-831 show a significant improvement for patients with mild and moderate Alzheimer's disease with a ADAS-Score 13 scale change of an average 4.1 as compared to placebo after 24 weeks of treatment, p-value equal 0.001. The CIBIC plus value shows 78% of patient improvement, p equal 0.01. NA-831 will well tolerate at 30 milligrams per day. However, for patients in the mild and moderate, the AD group report mild headaches and diarrhea. There were no serious adverse events observed. And as I mentioned earlier, we use the ADAS-Score as a benchmark. And in this particular graph, it's shown the ADAS-Score scale for the MCI versus placebo. You see on the graph, the blue line indicate the placebo, and the drug treatment was demonstrated by the red line. And over 24 weeks of study, the clinical improvement will demonstrate by the difference of the red line and the blue line represent the change of ADAS-Score scale over 24 weeks for patients with MCI. And it was about 3.4 for MCI patients. If you look at for the cohort of the Alzheimer patients, the change of ADAS-Score 13 were more profound. After 24 weeks of study, the ADAS-Score scale 13 show about 4.1 unit difference. And this is pretty significant for a Phase II clinical trials. Now, we have the benefit to compare our study with Biogen and lecanemab. We actually have a poster presentation and at the clinical trials on Alzheimer's disease CTAD conference on November 29 to December 2nd, 2022 in San Francisco. At that time, the data of lecanemab Phase III were presented at the conference, and we compare our ADAS-Score with ADAS-Score scale of lecanemab. And you can see that on the left side of the screen, the efficacy of lecanemab in Phase III, actually it was similar to the data found in their Phase II. And there was not a clear clinical meaningful benefit was established between the placebo and the drug based on phase 2 and even the phase 3. The changes over about several months after that is still the same as the 6-month time frame. The FDA mandate companies to use the 6-month data as the benchmark, and it could be extended as further as we wish. But usually, if the drug does not show clinical benefit in 6 months, it clearly does not help. The longer you take, you can see on their profile, the drug profile, lecanemab decline in pretty much the same pattern, the same kind of profile as much as a placebo. So the difference between the placebo and the drug, lecanemab, was not clearly significant. Whereas our drug NA-831, even over 24 weeks, 6 months, it showed a significant. And based on that, we continue to do phase 3, and I think we will have the result of phase 3, top-line result within the next year or so. I'd like to make a note that our drug NA-831 is quite different from aducanumab. One hand, aducanumab and lecanemab are monoclonal antibodies, require intravenous administration about once a month for the first 12 months. And the procedure requires expensive PET scan and quite painfully procedure of lumbar puncture as well. First, we are oral formulation based on neurogenesis, so we no longer need to measure PET scan or lumbar puncture procedure required. In terms of cost, aducanumab, AUHAM intravenous infusion costs about $28,200 per year per patient, plus about $46,000 for PET scan, lumbar puncture, and all the diagnostic procedure. The cost about $74,000 per patient per year. Whereas our treatment protocol is based entirely on oral formulation. Projected price about $12,000 per patient per year, with no PET scan or lumbar puncture procedure required. So at the present time, we work on the phase 3 clinical trials. We're seeking fast track designation, and we plan to have 2 programs, one phase 3 target treatment program for 475 patients with mild and moderate Alzheimer's disease over 52 weeks. And we also plan to launch a prevention program with 585 asymptomatic subjects. They're not a patient yet. Those are patient subjects without symptoms over 2 up to 3 years based on the guidance of the FDA. So this is a long-term study. We plan to have a patient enrollment in many sites in the US, in Australia, New Zealand, and other countries as well. With that, I'd like to stop here, and I'd be happy to answer the question you may have, and feel free to contact me. I'm with Biomed Industries, and my email here and my phone number here. I'm looking forward to hear from you. Any input, feedback are welcome.